Hello friends, welcome to the new chapter of cell biology. Myself Dr. Sunil Patil from Loknath Venkatrao Hiri College, Panchvati, Nashik. The chapter 10th, cellular aging and cell death. In this video we are going to see the cellular aging in detail. Cell aging, what is aging, what is cell aging, what is intracellular changes, what are the extracellular changes, what are overall changes. We are going to see in this lecture. It is video 12th of cell biology. I also make a PDF of the same. You can take it and for detailed study. So we are going to see the points from this chapter, the concept of aging theories, intracellular changes including free radical, the main part, and extracellular changes. So first we see what is the concept of aging. So aging is the pattern of changes that occur on as grows older. Gerontology is the study of individual and collective giving processes. Biological age, psychological age, social age, legal age, functional age. The definition: aging can be defined as the time related deterioration of the physiological function necessary for survival and fertility. Aging process is the process of growing old or developing the appearance and characteristic of old age. Aging is associated with changes in dynamic biological, physiological, environmental, psychological, behavioral and social processes. Definition of aging was proposed by Byron and Zerrit in 1985. The definition it is in this way: biological aging, senescing is the process of change in the organism which over time lowers the probability of survival and reduces the physiological capacity for self-regulation, for repair and for adaptation to environmental demands. So it is the scientific definition of aging or biological aging. So process of changes in the organism which over time lowers the probability of survival and reduces the physiological capacity for self-regulation, for self-repair and for adaptation to the environmental demands. So this is the definition. Now we are going to see the aging distinguished by modern gerontologist. Gerontology is the study of aging. Primary aging, which is postulated to reflect an intrinsic, presumably genetically programmed limit on cellular longevity. Primary aging seems to account for the relative constant maximum lifespan observed in almost all animal species. Secondary aging, so aging is divided into primary and secondary aging. Secondary aging is due to the accumulated effect of environmental insult, diseases and trauma. Secondary aging explains much of the variability among individual members of the same species. So primary and secondary are the two types of aging. So secondary aging, it is cumulative effect of the environmental insult, diseases and trauma, while the primary aging reflects the intrinsic factor, mainly genetic factor, then free radical, etc., genetically programmed limits, etc. So we are going to see theories of aging. Aging theories are mainly studied under these heads: organ-based theories, physiological theories and genomic hypothesis. There are various number of theories, but they are grouped into these three. The organ-based theories hypothesize that human aging results from incremental loss of organ functions driven by the immune system or alteration in neuroendocrine functions of the central nervous system. While the physiological theory, this group suggests that toxic level of cellular waste products accumulate over time resulting from radical damage, incapacitation of neuroprotective mechanism or some linkages of vital molecules, for example collagen, deoxyribonucleic acids and vital proteins, they get damaged. And third one, that is genomic hypothesis group, they hypothesize aging as a consequences of somatic mutation, multiple genetic errors or programmed cell death. Evidence that the hyphalic phenomena is under genetic control includes a fair of correlation between the doubling limit and the maximum species specific lifespan of the cell donor, a reduced doubling limit in cell culture from protein with genetic disease of accelerated aging. The precise mechanism underlying the observed limits on normal cellular division is not completely known, but several lines of evidence point to the telomeric shortening. We are going to see in detail what is shortening of telomeres, at least likely clock mechanism, and a consequential disarrangement exists between theories that human senses and brain alteration result from disuse versus overuse, use it or lose it theory. It is called as the disuse or versus overuse, and those that attribute aging to cumulative damage. Observe changes in the heart also, it is the result due to this. So we are going to see the primary aging structure and functional changes. Deposit of the aging pigment, that is lipofuscin accumulation, then the wall of the heart thicken and becomes stiff, the number of pacemaker cell decreases, and fatty and fibrous tissue increase around the SA node of the heart. These changes may result in slightly slower heart rate, heart beating decreases. A slightly increase in the size of the heart, especially the left ventricle, is common. The heart wall thickens, so the amount of blood that the chamber can hold may actually decrease. The heart may fill more slowly, so filling of heart is also decreases. To compensate, elderly subject demonstration doubling of percentage of the arterial contribution to filling it. Then theories of aging, there are various theories. I already told number of theories. They are grouped into 3 categories. We have already seen the theories are in this way: wear and tear theory, which includes the damage by overuse and abuse. The neuroendocrine theory, it is based on wear and tear theory. The genomic control theory, it is genetic inheritance theory. Then waste accumulation theory, limited number of cell division theory, cell division decreases. Then Hofflick theory, then death hormone, thymic stimulating theory, mitochondrial theory. The function of mitochondria ER get affected, it is main reason. Mitochondria are one of the easiest target of free radical, so this theory indicates that. Error and repair theory, then redundant DNA theory, then cross linkage theory. Old immune system are incapable to clear excess glucose in the blood, it is in the cross linkage theory. Autoimmune theory, you know the meaning of the word autoimmune. Caloric restriction theory, the rate of living theory, gene mutation theory, accumulation mutation theory, antagonistic pleiotrophy theory, disposable soma theory, order or disorder theory, the telomere theory, as we are going to see in detail. The free radical theory, we are going to see in detail these 2 parts. So there are various theories. So mechanism of cellular aging, genetic factor and environmental insults or environmental factor combine to produce the cellular abnormalities, which is characterized as aging. So genetic factor and environmental factor. Genetic factor, DNA repair defects, then various genomic abnormalities like IGF-1 pathway. The DNA repair defect causes the accumulation of mutation in the DNA, and genetic abnormalities, they are mainly the cellular signaling, they are abnormal, they contribute to the aging. Then environmental insult, including the free radical which causes damage to the mitochondria, ER, lipid, DNA. The reduced proteasomal activity, this togetherly causes the cellular aging. It is also additional replicative senescence and reduced ability to produce new cells. These things causes the cellular aging. So we have already seen the causes. Then hallmarks of aging, same thing. Genomic instability, it is one cause. Telomeric attrition, we are going to see telomeric attrition. Epigenetic alteration, then a loss of proteostasis, then deregulated nutrient senescing, nutrition. Mitochondrial dysfunction due to the free radical. then cellular senescence, then stem cell exhaustion, then altered intracellular communication. These are the hallmarks of the aging, overall aging. First, we are going to see the intracellular changes due to the aging. The nuclear pycnosis. With advanced age, the nucleus shrink and stain deeply. You can see this is the nucleus. It shrinks in size and stain deeply. It is the pycnosis. This is due to the condensation of the nuclear material. Aging is accelerated by chromosomal aberrations and somatic gene mutation. These are the reasons. Due to changes in the nucleus, degeneration of cytoplasmic organelle also takes place in the intracellular aging. The shortening of telomere, which is very important, accumulation of aging pigment, lipofuscin. It is lipofuscin accumulates, then accumulation of free radical, second thing. Third thing, weakened immune system, and fourth thing, decrease in rate of cell division. You can see these three things happen here. Lipofuscin accumulation at one end, then free radical attacked on that part, then there is weakened ecosystem. So you can see there is telomere shorten, eventually cell division get affected, it may stop, cell division may stop. So it is important effect. The telomere end which caps and protect the chromosome, it is get unprotected and it causes the different effects. In your syllabus, in our syllabus, it is given that study of free radical. So we are going in detail and deep in the free radical. A free radical is defined as any species that contain one or more unpaired electron occupying an atomic or molecular orbit. Unpaired electron, as they are highly reactive and unstable. They pull electron from other molecules and causes affected molecule to become free radical, and then new free radical pull electron to next molecule and a chain is produced. So example of free radical: superoxide anions, hydroxy radical, peroxynitrate, then hydrogen radical, nitric oxide, lipid peroxisome, peroxide. So normal cell, if free radical covers it, it shrinks, become irregular. The oxidative stress also causes this condition of the cell. So H2O plus the radiation is releases the free electron. This H2O become positively charged. Water molecule combines with other water molecule to produce the H3O and the OH negatively charged as a free radical. This OH free radical directly causes effect on the DNA. DNA produce the imbalanced proteins. So it is one of the effect. Then we are going to see in detail the what Dr. Herman studied that the postulate of free radical theory in 1956. The rate of living theory, which hold that lifespan is an inverse function of metabolic rate, which in turn the proportional to oxygen consumption. So it is inversely proportional to oxygen consumption according to the Dehnam-Herman. The Rebecca Gershman's observation that oxygen toxicity and radiation toxicity could be explained by the same underlying phenomena, that is oxygen free radical. So in the free radical, oxygen free radical, it is one part. According to Dehnam-Herman, cell continuously produce free radical by normal metabolism and oxidation of organic compound, and these free radical damage cellular macromolecule. It is normal process, continuously produce the free radical and which damage the cellular macromolecule, including the lipid, proteins, DNA, and due accumulation of this damage or this macromolecule over time period causes the aging. So it is important in the aging. We have already seen this free radical causes damage to cell. If there are antitoxicant, these antitoxicant neutralize the free radical and cell become protected. So we are going to see the mitochondrial free radical theory of aging or changes in mitochondria. This, according to the Denham Herman, modifies theory as, you have to understand this, about 95% of the cell is 95% energy of the cell is produced in mitochondria. We know this. Out of that, 97 to 99 of oxygen is used for this energy production. 97 to 99% oxygen which we take in, it is utilized for energy production, while 1 to 3% oxygen it is utilized to as a reactive oxygen species. So reactive oxygen species are formed. It states that the reactive oxygen species ROS are produced in mitochondria during the electron transport chain cycle, and they causes damage to macromolecule including lipid, protein, DNA of mitochondria. So lipid, protein, DNA get affected due to the ROS, means reactive oxygen species, which is only 1 to 3% produced in the energy production. So this thing happen, you can see here, instead of, you can see separately the excess oxygen, that is 1 to 3%, becomes superoxide radical, negatively charged, it combined with the water molecule H2O2, it forms the thiol oxidation, it causes cellular signaling, it is normal thing. But if it combined with the ferrous, it form the hydroxy radical. This hydroxy radical, it causes damage to the organelles from the mitochondria, the protein, DNA, lipid, macromolecule from the mitochondria. So this is mitochondrial free radical theory. ROS is formed, and this from the hydroxyl negatively charged free radical. Same thing is given here, the free radical from electron transport chain H2O2, and it may form the OH. Role of free radical, the oxygen mainly utilized for in energy production, but incompletely reduced 1 to 3%, it form the superoxide negatively charged combined with the water to form H2O2 hydrogen peroxide. There is Fenton reaction in the hydrogen peroxide which releases the hydroxyl radical, hydroxyl negatively charged radical. It is called the reactive oxygen species. ROS, reactive oxygen species formation, and which causes the damage. So pathological effects are due to the ROS and removal of free radical. The pathological effect, fatty acid to oxidation of fatty acid, generation of lipid peroxide, and disruption of plasma membrane organelle. So protein oxidation, loss of enzyme activity in abnormal folding. DNA oxidation causes damage to resulting in mutation and breaks in the DNA segment. So this is due to the ROS and in mitochondrial free radical theory. So same thing I have repeated third time. So reactive oxygen again same thing. Reactive oxygen species causes damage to DNA damage, RNA damage, protein damage, lipid damage inside the mitochondria. Then it results into the aging. So epigenetic alteration, due to this alteration in DNA methylation pattern, post-translation modification of histone, chromatin remodeling. In human and other mammals, DNA methylation level can be used to accurately estimate the age of tissue and cell types, forming an accurate epigenetic clock. A longitudinal study of twin children showed that between the ages of 5 to 10, there was divergence of methylation pattern in that twins due to environmental rather than genetic, as their genetic constraint is same. Then also there is divergence in the methylation, DNA methylation in the twin. So it is proved that it is due to environmental factors or environmental insult. There is a global loss of DNA methylation during the aging. So study of free radical, the cellular sources of free radical are mitochondria and endoplasmic reticulum, where free radicals are produced. Then in the protein synthesis, in the energy production, the enzymatic sources, cytochrome oxidase, then xanthine oxidase, and membrane NADPH oxidase. So environmental sources, ionizing radiation pollution, Then ROS, you know the meaning of ROS now. It causes in the DNA breaking of strand, then depurination and depyrination, then mutation of bases, then protein DNA cross linkages are formed. In lipids, enhanced membrane fluidity and permeability, breaking of lipid chain. In protein, modified amino acids are formed, breakage of the peptide chains, increased proteolysis or degradation, inactivation of enzyme. So this thing happened due to the ROS. So in the endoplasmic reticulum, oxidation of toxic compound normally takes place. The foreign toxic compounds due to the in presence of cytochrome P450 or superoxide radical. Due to superoxide radical, they become less toxic compound normally, but free radicals are formed due to enzymatic sources. Cytochrome oxidase add 4 electron onto molecule of oxygen in a series of reduction reaction. So in this way, xanthine to uric acid to allantoin in presence of these enzymes. Each of this reduction reaction may potentially have superoxide radical as a byproduct. This superoxide radical causes the ROS. Environmental sources: exposure to ionizing radiation like gamma rays, X-rays, UV rays excited the electron in many atoms, thereby generating large amount of free radical. Oxygen free radical in the atmosphere considered as a pollutant. Interaction with chemical automobile exhaust fumes, smoking of cigarettes, cigar or BDs, they causes the oxygen free radical in the environment. So H2O become hydroxyl negatively charged hydroxyl radical, which is negatively charged in presence of UV rays. Water become this, and it causes the free radical. How free radical causes aging? The radical formed by metabolism, by irradiation or by toxic agents causes the cellular damage, aging and death by different routes. A DNA altered, there is RNA alteration, there is a lipid peroxyl of alloxyl radicals are formed, altered protein and enzymes, they causes the impairment of different things, organelles, and excess of specific proteins are for absence of specific proteins. Due to this, the cellular damage, aging and cell death occur. So free radical affects 3 component of cell: lipid degradation, lipid peroxidation, then biomembrane and subcellular organelle are partially sensitive to oxidative attack due to the presence of polyunsaturated fatty acids PUFA in their membrane phospholipid. So you have already studied this. DNA damage in this free radical can damage by following ways. So the first way is the nitration, guanine into 8 nitroguanine, guanine into 8 nitrooxidoguanine. So inter and intra strands cross linkages are formed. There is formation of inter-intra strand linking formed due to the covalent bond formation between opposite or same strand DNA strands. Covalent bond is formed between second nitrogen in one strand with the second carbon of another guanine located in other strand or opposite strand. So direct effect, indirect effect on the DNA. Double stranded breaks in both cases. Free radical also causes same effect. Single strand break, double strand break due to the radiation. Protein degradation. Protein degradation occurs due to in the cell damage. The protein degradation in this way occur due to ROS mainly or hydroxyl radical. The free radical in this way, we have already studied DNA degradation, protein degradation, lipid peroxidation. It results into cancer, heart attack, muscular degeneration, atherosclerosis, pulmonary disorder, retinal damage, asthma, hyperoxia, and it causes the aging. So these are effects of these free radicals which result into aging. So what is antitoxicant? Any synthetic or natural substance that delay or inhibit oxidative damage due to the free oxygen to a target molecule and capable of neutralize free radical by donating electron or hydrogen molecule. They are called the antioxidants. They prevent cell damage and tissue damage and acts as scavengers. So unpaired electron and antitoxicant donate electron, they become saturated. So antitoxicant causes antitoxicant enzyme include antitoxicant vitamins. The antitoxicant enzymes superoxide dismutase, it catalyze the rapid dismutation of superoxide radical to hydrogen peroxide and to oxygen. So in this way hydrogen peroxide and oxygen are formed. Same thing is shown here. The conclusions: free radical theory of aging is only concerned with all free radical in our body causes cumulative together damage which eventually lead to loss of functionality and ultimately cell death. This theory is supported by vast number of experiment and observation. This provide important view about free radical. So we are continuing the conclusion of free radical theory. There is no doubt that reactive free radicals such as hydroxyl or peroxy radical can damage biomolecule and their activity can be suppressed by antitoxicant free radical scavengers such as alpha tocopherol, that is antitoxicant, vitamin E, and ascorbic acid, vitamin C. They act as an antitoxicant to neutralize the free radical. But this theory could not find differences between various free radical. However, many free radical help to regulate many functions in cell, like cell signaling, means cell signaling, killing of bacteria, cofactor of enzyme. Free radical theory aging must be broken, broadly studied. It is need to study broadly. Extracellular changes, the changes occur in the, now we are studying extra, up to this we have studied the intracellular changes, the changes outside the cell. The changes occur in the intercellular spaces and in the lumen of blood vascular system are called as the extracellular changes. Intercellular spaces. The dementia is a serious loss of cognitive ability. Alzheimer's disease, it is one example, you know very well. Alzheimer's disease, a substance called amyloid built up and causes the early death of brain cell. In Alzheimer's disease, there is cell death in brain which result in a progressive loss of memory and brain functions. Then atherosclerosis, it is one effect. In the extracellular effect, you know the diseases of bone marrow and bone. Fibrosis is formation of excess fiber connective tissue in an organ tissue. This is an opposed to formation of fibrous tissue as a normal constituent of organ. Cirrhosis of the liver, it is a common example of fibrosis. The Parkinson disease. So Parkinson disease, these are the effects of Parkinson, the typical appearance of Parkinson disease: masked facial expressions, forward tilt, reduced arm swing, then slightly flexed hips, etc. Changes in collagen, they are important for us for the theory. Extracellular changes, there is an increase in the amount of collagen protein deposition in the intracellular spaces. This influences the permeability of cell membrane, affect the speed of diffusion of substances in and out, significantly influence the process of aging. The wrinkles and aging. Wrinkles are byproduct of the aging process. With age, skin cell divide more slowly and the inner layer called the dermis begins to thin. Dermis become thin and thin. The network of elastin and collagen fibers which support the outer layer with the age Skin also loses the elasticity, is less able to retain moisture. Oil secreting glands are less efficient, and slower skin, slower to heal. So these are effects of the extracellular effect of the aging. The stem cell exhaustion, the decline in the regenerative potential of tissue is one of the most obvious characteristic of aging. Hematopoiesis declines with age, resulting in the diminished production of adaptive immune cells, and increased incidence of anemia and myeloid malignancies. So stem cell exhaustion, so these things happen: osteoporosis, anemia, the decrease in repair of muscle fiber, decrease in intestinal function, decrease in fracture repair, etc. The hematopoietic stem cells, mesenchymal stem cells, and intestinal epithelial stem cells and satellite cells. Altered intracellular communication. Beyond cell autonomous alteration, aging is involved changes at the level of intracellular communication. So it results in the neuroendocrine dysfunction, inflammation, then immunosenescence, then bystander effects. Functions of interconnection between the hallmarks of aging. We have studied the hallmarks. Primary hallmarks including the genomic instability, telomere attrition, epigenetic alteration, loss of proteostasis. Then antagonistic hallmarks including the deglutition, nutrient sensing, mitochondrial dysfunction, and cellular sensing. Integrative hallmarks include stem cell exhaustion and altered intracellular communication. We already seen this, but they are grouped into these three: primary hallmark, antagonistic hallmark, and integrative hallmarks. So intervention that might extend human health span. So stem cell based therapies, these are interventions, telomere reactivation, etc. So this is about the cell aging. I think you got everything. If you have any doubt, you may directly contact me. On the scrolling below the video, my email is given, so you can contact by the mail. So you may search BioEduBit in the YouTube channel to get all the videos. Have a nice day. See you soon. Thank you.